All right. How are we doing today?
I'm just going to ask, is the substrate level phosphorylation, is it the step or the enzyme?
It describes So, test tomorrow. Scale of 1 to 10. How are we feeling about it? We are all over the place. We got some 2s, a couple 10s maybe, some 4s. All right. So we are going to be covering what? We have the end of chapter 4, 6, 7, 8, 9, and 10, right? So we got some work to do today. I'm going to try to get through all of that, and then if we have time at the end, do you all just want to go through that mock exam that I made? Cool. Okay? No more than 2 hours. No more than 2 hours. 2 hours is my cutoff. I got to go study myself. All right? No, I don't study. Okay, I will not study for boards, and I will fail them, and I will just stay tutor at Parker forever. That sounds great. Yeah. All right. Before we start, I was studying before this, like I should be, and we have farm talks, and in one of our tests, we had to describe the mechanism of action of cortisol, right? And so I was like, I feel like I should know this answer, and then I was like, oh yeah, like cortisol is a nonpolar substance, so it just like diffuses through that cell membrane, and then goes and diffuses through the nuclear membrane, and then attaches right onto DNA, causing the expression of gene expression, creating proteins, right? And that was like the practice test question, and I was like, okay, cool. Like there's time. So there's tie-in later. I promise. It might be until try 5. But there's tie-in, I promise you. Yeah. All right. So do we start on page 60? Yes? Or is Lineweaver on there too?
No, we did that.
Lineweaver was on the last one. Cool. Started effects of pH. Cool. pH affects enzymes. That's all we need to know, right? Structure determines function. What's the body's pH? 7.35, 7.45, right? So if the body's in like the blood circulation, it needs to be that enzyme. But if it's in the stomach, right, where HCl is secreted, and we have, for example, pepsin, or pepsinogen, which is turned into pepsin because of HCl. So it's working in that superacidic environment. So that enzyme has to be within the pH that it's designed to be in for it to work. That's all that's saying. Won't work if it's outside the pH range. All right? So we have like pepsin. Here we go. Here. Pepsin works in low pHs, whereas glucose 6-phosphatase works in, I guess, a more alkaline pH. Temperature is the same thing. Most of the enzymes that in our body are going to be at body temperature, right? If they're not at body temperature, not good. That's why we have a fever, right? If we have a fever, that's our body's natural response to bacteria in our body that shouldn't be there, right? So we increase our body temperature, denaturing proteins in that bacteria, therefore allowing that bacteria to die. So fever equals good, right? Did y'all learn that in philosophy yet? Fever equals good. Different types of inhibition. 3 different types of reversible inhibition. Need to know it for sure on the test. We have competitive, uncompetitive, and mixed. Competitive, the substrate, the thing that the enzyme's working on, is competing with that binding spot with an inhibitor. All right, they're both trying to bind to the exact same spot. Okay. Whoever gets there first wins, all right? So if the substrate gets there first, great. Enzyme's going to work, convert that, and make a product. If the inhibitor gets there first, no product, right? Turns it off. We can override the likelihood of the inhibitor binding by increasing the amount of substrate. Okay? So if we just threw in a bunch of substrate, the likelihood of it reaching that enzyme before the inhibitor goes up because there's more in there, there's more proximity, it's a better chance of it happening. And what we need to know here is that the Vmax remains unchanged, but the Km increases. All right, I looked up like 4 videos today trying to find someone to explain that really well, and no one did. So please just know that with competitive, the Vmax remains unchanged, the Km increases.
A quick little explanation for it is basically just that it's not affecting the enzyme's capacity. So the enzyme can still work just as fast. So it's not going to change the highest velocity that you can hit, but you need more substrate to overcome that. So your concentration is being affected, which affects Km. So you have to increase the substrate, but the enzyme's capacity for speed isn't affected. That's how I process it.
Cool. I would give you the mic to drop it, but I like it on me. Good. Did y'all hear that? Question? There probably won't be a graph on there. It'll probably be like in a word form. He'll probably make it a word problem where the Km increased or the Km decreased and the Vmax stayed the same, and you'll have to determine which one it is. So based on the context clues. All right. So what she's saying is that the max speed of the enzyme doesn't change. But the Km increases because we can increase that substrate concentration, right? We can increase that substrate concentration to override the inhibitor, making that Km increase, but the enzyme can still function as fast as possible as need be. We're not changing the enzyme at all. That sounded less good than what she said. If you're listening to the podcast, listen to what she said. Uncompetitive, just know Km increases, Vmax unchanged. There's a box at the end. It's all right there at the end. Uncompetitive. The inhibitor affects the enzyme's catalytic function, but not the substrate binding. With this, the substrate has to bind first. I talked with Dr. Sarkar that the substrate has to bind first before the inhibitor can come in. Just so you know that. We're not going to use my vuvuzela example because that did not go well. The inhibitor is going to bind at an allosteric site. Okay, with competitive inhibition, the inhibitor was binding at where the substrate binds. With this one, the inhibitor is binding at an allosteric or a site other than where the substrate binds. Okay, and it can only bind to that enzyme substrate interaction where the enzyme's already bound. With uncompetitive inhibition, we have our Vmax decrease and our Km decrease as well. How I remember that is, I think of like a U, right, or U, and I just draw arrows down on both. So both decrease with that U. And I redrew that. Oh, yes, I left the pencil here from like forever ago. All right. So like a U. Oh, this pencil's not mine. Whatever. Both can go down. All right? Both can go down with uncompetitive. That's what you need to know. The substrate is free to come and go as it wants, but once that inhibitor binds, that enzyme shut off. Okay? Lastly, non-competitive or mixed. The inhibitor can bind at any time. Okay? Inhibitor can come and it can bind at any time at an allosteric site. This one's also allosteric. And that doesn't matter if the substrate is there or not. We are going to decrease our Vmax, and our Km is going to be unchanged. Okay, I'm sorry to say, just memorize it, or listen to what she said earlier. Actually, let's say it again. We're going to give you the mic.
Different.
This one's different.
This
Can you
one's
explain
different.
it? Put you on the spot.
Okay, hold on, just a second. Let me gather my thoughts. Okay, so.
Or explain the first one again, and then.
Okay,
you can-
so for competitive inhibition, you're not changing the capacity of the enzyme. You're just filling that spot with another thing. So the capacity or the speed of the enzyme stays constant, so you don't change Vmax, but you have to add more substrate so that you don't block the site. So you need more substrate, and Km is what deals with substrate. So the Vmax will stay the same because you can still do it just as fast. You just have to have more substrate to make that happen. And then with uncompetitive inhibition, let's see. So Vmax decreases because you're affecting the enzyme's catalytic function. So if you're changing the speed that the catalyst can work, then the maximum speed that it can hit is going to be lower. And since you're lowering that, it's going to lower the Km because if it can only work so fast, then you're needing less substrates to add into it. And then, let's see. This one. So, I'm not actually sure what the explanation for that one is.
We can interpret for the next one. Give her a round of applause. She's going to get 2 dum-dums for that. 2. And I'm going to throw them one at a time. That was high. That was also high. All right. Chart has all that information right there. Irreversible inhibition, covalent bonds, right? Covalent bonds are strong bonds, very difficult to break. So those are going to be permanent changes to that enzyme, turning them off. Okay, examples here are going to be aspirin and the cyclooxygenase pathway. You'll learn all about that in Path, but we'll get to y'all's Cox blockers and try 2.
I do actually get this now.
Cox blockers, COX. Get your minds out of the gutters, all right, y'all. Come on.
I do get the third one now, though.
You do get the third one?
Yeah, because with uncompetitive inhibition, you have to bind the substrate, and so the Km decreases. But since the substrate can bind at any time, it's not affecting concentration of substrate, and Km is dependent on concentration of substrate. So since it can bind at any point during the process, it's not going to change. That's better.
Woo. Okay. Probably going to be at least 1, maybe 2 questions on that on the exam. So irreversible covalent bonds, aspirin, cyclooxygenase enzyme. Moving on, irreversible, not that big of a deal. Protein-protein interactions. The proteolytic chain cleavage is irreversible, and pretty much that's addition by subtraction. So some enzymes are activated when peptide segments are removed. So taking off that protein is what turns that enzyme on, and that's irreversible, so addition by subtraction. Modulator binding, it's the modulator. What happens is it's going to bind to the enzyme. When that modulator binds, it's going to change that enzyme's conformational shape, making it to where that substrate can bind to it. All right, so that modulator kind of turns it on. It's like using interferential on a patient, right? So if you have a patient that's really locked up and you're having a hard time adjusting them because they're just so tight, so you put that e-stim on them, you put that interferential on them to relax their muscles, making it to where their body can better receive that adjustment. Something similar like that.
So it's more like allosteric stimulation.
Yes. Yes. Feedback inhibition, that's when the end product inhibits the beginning product, right? So once we've come to the end, we don't need to do that process anymore. So that last end product goes and turns off the process at the beginning. Feedback inhibition says we're good. Different types of reversible covalent modification. he could pick any one of these, but just know that these are examples. I think when I took the test, it was which of the following is an example of reversible covalent modification. I asked to see the test, but on Thursday of last week, but he hadn't made it yet. So I think he made it on Monday. Look at these two, regulation by proteolytic cleavage, just in case. So trypsinogen to trypsin, that happens in our stomach, and chymotrypsinogen to chymotrypsin, just in case he says, which of the following is a proteolytic cleavage? You never know. You never know. Half of a chapter down. 5 more to go. 5 more to go. Insulin, glucagon, other hormones. Major nutrients of our bodies, carbs, fats, proteins, right? We know that. Here's this nice little chart. We've seen this before. We know this chart. I talked about how cortisol was a steroid earlier. Glucose is a metabolic necessity for all nutritional states. Glucose is our most important sugar, okay? Our liver is the major tissue for fuel metabolism. In lab dye this try, we are talking about different types of tests to see which organs in the body may or may not be working very well. And like liver, there's like 4 different tests for the liver. And it's so important with regulating that fuel metabolism and also with a lot of protein creation as well. So liver, huge, huge, huge. When in doubt, put liver, okay? Acetyl-CoA is the thing that connects all our carbs, fats, and proteins, all right? All 3 of those can make acetyl-CoA. So it's kind of the center of the universe, or it's the universal currency of our energy needs.
Andrew?
Yes, sir?
Those 3 make acetyl-CoA or acetyl-CoA or both?
Both.
You can go either way.
It can go any way.
Carbohydrates could make acetyl-CoA or acetyl-CoA could make carbohydrates.
Mm-hmm. Both ways. All right, this picture, don't worry about it yet. Not that big of a deal. It just shows that we can make acetyl-CoA from everything. Pancreatic hormones, insulin and glucagon. Okay? We have our adrenal hormones, epinephrine, norepinephrine, cortisol. Y'all will learn more about that in future tries. Whatever. Insulin, glucagon control our blood glucose levels. Okay? If we have too much blood glucose, the body is going to secrete what? Insulin, right? Diabetics. That's what we initially think of. Right? Type 2 diabetics. They can't control that blood glucose level. If there's not enough blood glucose, then we're going to secrete glucagon, right? Glucagon's going to take blood glucose from the cell, release it into the bloodstream. We need to know the different types of state that the body would be in to have those be released. So insulin is going to be released if we have too much blood sugar, right? So it's going to be like after a meal, so like in our fed state. Okay, we've got too much sugar, we got to take it out and store it somewhere. All right, but if we're secreting glucagon, that's when we're fasting. Okay, that's in our fasting states. We don't have enough blood sugar because we're not ingesting it, so we need to take it out and put it back into the bloodstream where it's needed. Does that make sense? Cool. Cortisol mediates long-term stress. That's why the adjustment is so powerful, because we can decrease cortisol levels in the body. It's wonderful. Glucagon, insulin are antagonistic to each other, so they are opposites. Pancreatic beta cells secrete insulin. Pancreatic alpha cells secrete glucagon. Need to know that. Okay? Like I said, insulin counters high blood glucose. It's taken up and stored. Glucose is taken up by the muscle and fat tissues, where they're converted to glucose 6-phosphate, right? First step in glycolysis, hexokinase. Who all is going to do the challenge? Raise your hand. Who here is going to brave themselves and do the challenge after this? We got 3 hands. I like it. The enzyme speed challenge, I posted it on Facebook. All right, we'll talk about it at the break. I like it though. Insulin secretion. We need to know the order of insulin: pre, pro insulin, mature insulin. Mature insulin is the one that's active, that's usable. These other states are inactive, and we kind of cleave other parts of it to get to that active state. Just know the order as far as I'm aware. And it makes sense. So pre, pro, mature. Insulin activates the oxidation of glucose in the liver for energy, glycogen synthesis to store energy, and fat synthesis. Why that is if we have glucose in the blood, we're going to break it down, try to get the energy from it. Or if we have too much, we're going to store it as glycogen. And if we also have too much, we're going to store it as fat too, right? If we overeat, we're going to put on some weight. Metabolic effects of insulin. Insulin increases glucose uptake. So we're going to make glycogen from that uptake. We're going to stop breaking down glycogen because we're trying to make it. We're going to go through glycolysis to make energy from it, right? We're eating to get energy for us to keep living and doing things. We're going to increase increase fatty acid synthesis in the liver, right? If we're eating too much, we're going to store it. And triglyceride synthesis in the adipocytes. And glucagon is the exact opposite. All right, this is kind of where that test question is going to come from. He's going to give you an example here, and you're going to need to know where, what's being secreted, or what kind of state you're in, based on if it's insulin or glucagon. That test question would be like, in glucagon, what's happening in the body? And he'll have like increased glucose uptake, increased glycogen synthesis. What's the other one? Glycogen synthase activity, so that's like breaking down glycogen, those types of things. Insulin is a tyrosine kinase type receptor. I learned that in chapter 5 briefly. Glucagon is a G protein coupled receptor. It shouldn't get much more deeper than that. Low levels of blood glucose, once again, glucagon, it's also going to decrease insulin release, right? We don't want to have those competing at the same time. That would be a waste of effort and energy and time. We know how G protein coupled receptors work. Here's an example of feedback inhibition. Just read that on your own. Chapter 7. Questions on glucagon, insulin. Do we know insulin, high blood glucose, like fed state, glucagon, low blood glucose, fasting state, insulin, beta cells, glucagon, alpha cells, insulin, tyrosine kinase, glucagon, G protein coupled receptor. Those are the big deals. Those are big deals. Bioenergetics, our favorite chapter. Gibbs free energy. We need to know the definition of Gibbs free energy. It's available energy that can be converted to do work, right? Something that we can use. If we are breaking bonds or we're releasing energy, that's an exergonic type reaction, right? Energy is exiting. If we are making bonds or going through anabolism or adding things together, that is an endergonic type reaction, right? And endergonic reactions and exergonic reactions, all of those combined make our metabolism. All right? So people with high metabolism, they just have more things being broken down than building up. Free energy, useful energy. Delta G is our main thing that we're using. Need to know our different laws of thermodynamics. All right? He could easily ask you, what is the first law, and you have to give the definition, or he could give you the definition, and you have to say if it's the first or second law. First law, energy can be neither created nor destroyed. It's that simple. You can change the form of it, but you can't make it or create it, right? Whatever innate intelligence did, that's there. We can just use it. Okay, second law, the total entropy of the system must increase for a process to occur spontaneously. So entropy is the extent of disorder or randomness in a system, right? So that's like when we give off heat, when we're exercising, right? That's a type of randomness, always increasing, okay? Entropy, big thing here is just know this equation. Delta G is equal to delta H minus T delta S. Delta S is your entropy. Delta H is, he's not going to ask you what those mean. He's just going to want you to know, do you know what this equation is, as far as I'm aware.
What's that equation called?
Just an equation relating delta G to enthalpy of a system. But just know the equation. Information from delta G. We can get 3 things and inadvertently 4 things. First is whether a reaction releases or requires energy. So if our delta G is negative, does it release or require energy? Release. Boom. All right, done. We got that one. The delta G is the amount of energy involved in the reaction. So that's just if it's a big number or it's a little number. And 3, the ratio of the concentration of the products and the substrates at equilibrium. And that's going to be with this equation down here. But we're not going to have to, you're not going to have to do any science, any math. You have to do any math. You might have to do a little science. You're not going to have to do any math on this exam. One thing I want to point out is that it says nothing about how fast that reaction is going to happen. Okay? It tells you nothing about that speed. Every test I've seen, that's been one of like the answers. Not one of the answers, but one of the like trick you answers. So like if you have a delta G of like negative 1,000, whatever, you'll be like, oh yeah, like that happens really fast. No, it doesn't. It just tells you that it's going to happen. Okay? There's processes that are going on from like millions of years ago, still happening right now, but it's going to finish eventually. It tells you nothing about how fast it's going to go. Know this equation as well. Delta G is equal to the negative RT natural log of KEQ. Our KEQ is just products over substrates. Okay? Know that equation. And I'll use, thank you, the science of chiropractic. All right, significance of delta G values. I looked it up finally. That equation that we just did, negative RT natural log of KEQ. Okay? That graph looks like this. If you plug that into your TI-80 whatever calculator, that's what you'll get. So this is 1 here. So this is 1. Keeping this in mind, when KEQ equals 1, the delta G is 0. So KEQ is on our x-axis. Delta G is on our y-axis. So when KEQ is equal to 1, delta G is equal to 0. It's just plugging it into the equation. All right? So anything greater than 1, delta G is going to be negative. All right? Anything less than 1, delta G is going to be positive. So if you can visualize that graph or just draw that graph on the test really quickly, that should be a pretty simple answer to get right. Okay, so that's why we have this information. That's where these values come from, is based on that graph of that equation. Does that make sense to y'all? Okay. If delta G is negative, we've already talked about it, exergonic. If it's highly negative, it's going to go to completion and is irreversible. Doesn't tell you how fast, though. Just says it's going to go to completion. If it's positive, it's endergonic, so it's absorbing heat, it's making bonds. And if it's greatly positive, if it's too high, we don't have that much energy, so it's just that system is going to stay the same, right? It's trying to wake up at 5 a.m. to go run a marathon, all right? That's just too much energy to do. We're not going to do it, so we're just going to turn our alarm off and go back to sleep. System's going to stay the same. And delta G is equal to 0. There's no net change taking place. We're at equilibrium, right? We're neither making nor breaking bonds more one way or the other. Endergonic reactions proceed by coupling to exergonic reactions. Okay, that's big, 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 big. So the exergonic reactions release the heat, endergonic picks it up, or the energy, releases energy, endergonic picks it up, makes the bonds, or does whatever it needs to with that energy. All right, know that exergonic gives it off, endergonic picks it up, not the other way around. And just here's an example. The exergonic reaction is A going to B, and the endergonic reaction picks up that energy and turns C into D, and then there's our entropy right there in heat, right? The entropy is always increasing. The disorder and randomness is always increasing. Is that law number 1 or 2? 2. Good. If we're making bonds, that's anabolism. We're adding things together. If we're breaking bonds, if it's exergonic, that's catabolism. It's a catastrophe. Things are breaking. Okay? Things are being broken down. High energy phosphates, high energy phosphates play a big role in energy capture and transfer. All right, that's why ATP is so huge. That energy stored in those phosphate bonds is a lot, and we can do a lot with it. So that's why it plays a huge role in us trying to make it with glycolysis, because then we can break it later to do work within the body. And so we hydrolyze ATP to release energy, yada, yada, yada. We can make ATP with these processes. Here's another example of coupling exergonic reactions with endergonic reactions. Maybe start with reaction 2, though, because it's the exergonic one, because it's negative. So we hydrolyze ATP, giving off energy. Couple that to putting that phosphate onto glucose, making glucose 6-phosphate. And the net reaction is still negative, releasing energy. Yeah?
So if you had those reaction 1 and 2 combined together and it's still endergonic, would you need a third reaction that was higher exergonic to make it proceed?
Most likely, yes. Most likely, yes. Other nucleoside triphosphates, right? So we have ATP, we know that one, but we also have UTP, GTP, CTP. I put that on y'all's mock exam, right? It's just, did you look at this last page or not? It's all the ones that are in DNA and RNA, right? So it can be G, C, A, T, or U. Uracil for RNA. So when in doubt, just try to fall back onto those from your biology. Questions on that chapter? It's not too bad. Just know the basic concepts so you can apply it to whatever concept he tries to pull you up.
Isn't the formula to find delta G is G1 minus G2 equals delta G?
Correct.
How does that work in that last reaction?
You add the negative, or use. That's going back to like.
Because it should be negative 10, shouldn't it? Or negative 11?
Oh, did I hear that? No, G1 minus G2 is delta G.
These are both delta G's. So you add the delta G's together. The one you said is just defined delta
those are both. Yeah, you're right. They're both delta G. cheese, not cheese.
It's not a cheese.
Yeah.
That makes sense.
Those are details like I don't think he's gonna like have you adding delta G's together. Also, that's just conceptual. No math. No math. Introduction to carbohydrates. Carbohydrates have carbons, hydrogens, and oxygens in them, right? Glucose is the most important carbohydrate that we have. It's our major fuel. That's why we go through glycolysis. That's why it's named after it, right? It's named after glucose. We can store it as glycogen. We can make nucleic acids with it. We can use it for cellular recognition, lubrication of joints with our gags, signaling molecules, and we can have structural elements as well. All right, very versatile. Glucose, very, 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 very important. Right? That's why insulin and glucagon have such a huge role in making sure that we have enough of that in our blood at all times. Different types of carbohydrates. They're broken up into aldehydes, ketones. Fructose is an aldehyde or ketone? Fructose is a ketone. All right. Ribose is it an aldehyde or ketone? Ribose. Aldehyde. Good. We'll get to that rule in a little bit. Monosaccharides are the basic unit of all carbohydrates. It's just like amino acids are the basic units of proteins. It's that same relationship. Disaccharides are two monosaccharides. Pretty simple. The one here that's tricky that he could ask you is oligosaccharides are 3 to 6 monosaccharides. Polysaccharides are greater than 7. All right? Just got to know those numbers. Monosaccharides, either aldehydes or ketones. And aldehydes or ketones, to give you an example, aldehydes over here with glyceraldehyde, so triose. That carbonyl group is on the end here, that C double bond OH, whereas with our dihydroxyacetone, which is a ketone, that aldehyde is in the middle. So that C double bond O is in the middle of the molecule. It's just talking about where that's located at within that chain. And, so an aldehyde is classified as an aldose when it's talking about sugars, ketones, ketose, pretty self-explanatory. And we can divide those into triose, tetrose, pentose, hexose, or above. Okay? And when we're talking about this, triose, we're talking about the number of carbons that are in that sugar. Okay? So a triose has 3. Tetrose has 4. I'll stop there. All right? We need to know these, all right? We need to know both whether it's an aldehyde or if it's a ketone. And or, actually mostly and. So our trioses are glycerose, dihydroxyacetone, right? We see that with glycolysis, with aldolase, creating glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, right? It takes that 6-carbon, fructose 1,6-bisphosphate, breaks it in half, making 2 3-carbon trioses, right? So glyceraldehyde or glycerose, dihydroxyacetone. Pretty simple. Tetrose are 4 carbons, erythrose, erythulose, and we'll get to that rule later. But big ones, glucose, fructose, I would say, knowing that fructose is a ketone. Stereoisomers, compounds that have the same structural formula but differ in their spatial arrangement. Okay? Dr. Sarkar, when I talked to him, he said mirror images of each other. So whenever you see stereoisomer, you're thinking mirror images. Asymmetric carbons. What's another word for an asymmetric carbon? Chiral, right? Optically active. If we have asymmetric carbons on a molecule, we can make isomers. And isomers are pretty much just glucose versus fructose, right? They have the same chemical formula. They're just arranged in different ways. All right, whether that carbonyl group is here at the top or it's here in the middle. All right. We can determine the amount of isomers, like for example glucose has, by the equation of 2 to the n, where n equals the number of asymmetric carbon atoms. So for glucose, if we look over here, he has it written out somewhere. Here we go. Nope. Glucose, the ones in red are the asymmetric ones. So we have 1, 2, 3, 4 asymmetric carbons. That carbon is binding something different every time. So we just do 2 up to the 4. So glucose has 16 different potential isomers. And I think that was the example he used on our exam.
You know what I was studying with Brittany, actually we're going over this, and so just like side note for the test, if it's a number larger than 16, just automatically mark it off, because it's not going to be a number larger than 16. That's the highest it can go up to.
Cool.
2 to 4.
Yeah, and yeah, don't pick anything higher than 16. Good. She was saying don't pick a number higher than 16 on the exam. It's probably going to be too high. And why is it going to be too high?
Because 2 to the 4 is 16, it can't be more than 4, because that's what's in the book. That's all we need to care about.
Is there more than 6 carbon atoms?
Not that we're dealing with. So she's saying the biggest molecule that we have is a 6-carbon molecule. So we can't have more than 4 carbons. So the highest that we could go to is up to 16.
So aldehyde and dihydroxyacetone are isomers, but isn't the dihydroxyacetone
Say that again?
So these two are isomers, right? But isn't this symmetrical? I just the exception to the rule.
Don't worry about it.
Okay.
Don't worry about it. D and L isomers, it's the same thing that it was with proteins, right? What side the OH group is on, which way it reflects light. D, dextro to the right, L, levatory to the left. The biggest thing with that is only D isomers are used to synthesize carbohydrates in humans. All right, it's big, bold, top of the page. Don't miss it. D isomers, your carbs. I thought of that like desserts, right? Desserts have a lot of sweets, so are D isomers. Proteins are proteins D or L? L, right? So they're different. All monosaccharides are asymmetric except dihydroxyacetone. I got you. All right, so all that's saying is we zoom in really close. That's zooming out. I'll get that right eventually. That's really close. For this one right here, we can draw like an imaginary line through it to where that side is the same as that side. I don't think he emphasized it too much, but it's the exception. Let's zoom out. Let's do that. All the other ones, if you drew a line through them anywhere, they would be different on both sides, asymmetric. Enantiomers are optically active isomeric forms that are non-superimposable mirror images. So stereoisomers, mirror images. Enantiomer, non-superimposable mirror images. Associate those two together. Okay? You cannot put one on top of the other. It's like your left and right hand. You cannot make your left hand your right hand no matter which way you turn it, right? No matter which way you turn it, your left hand will never be your right hand. Don't do this because you'll just confuse yourself. Just do this. Okay? D isomers, naming of aldehydes versus ketones. All right, the rule is if it ends in OSE, it's an aldehyde. Okay? If it ends in eulose, it's a ketone. The exceptions are glyceraldehyde, but it tells you, which is really nice. Fructose, which I've already drilled into y'all. It's a what? Ketone. And dihydroxyacetone, which is just the opposite of glyceraldehyde. So you already know that that's a ketone, 3-carbon ketone. Good. So everything else follows the os versus eulose rule. Our 4-carbon is erythrose, right? 5 carbons, ribose, arabinose, xylose. I remember, I guess you kind of could do the same thing with ribose, but xylose, there's like 5 points on the X, so it's a 5-carbon sugar. You kind of do the same thing with ribose, kind of make the R with 5 points. It'll look more like a K, but use your imagination. And then arabinose is obviously going to be in the same category as ribose, right? 6 carbons, glucose, mannose, galactose. Those are our aldehydes. Okay, so glucose is an aldehyde. 3-carbon ketones, dihydroxyacetone, right? What enzyme makes dihydroxyacetone in glycolysis? Aldolase, right? Aldolase. Sucker. Don't be a bad throw. Hell yeah, that's what I'm talking about. For once, I'll take it. All right, dihydroxyacetone. That's how he's going to ask a question, like what makes this, and you have to just be able to put yourself in that part of glycolysis right then and there. The more you look at it, the faster you'll be able to do that. Erythrulose, right? Erythrose was 4 carbons, so is erythrulose, but this one's a ketone. Ribulose, xylulose, and fructose. All right, it's that simple. Those are the only ones you need to know. If it's not underlined or boxed, don't worry about it. Okay? Epimers are isomers differing as a result of variation in the configuration of our OH and H groups on carbon atoms. All right? And specifically, we're talking about epimers of glucose right here. Glucose is our most important sugar, right? Bless you. It's our most important sugar, so we're trying to see how things differ from glucose. We have 2 examples here. We have mannose, where the 2nd carbon hydroxyl group is on the left instead of on the right. So we're just looking to see where it's wrong. This one's on the left over here, so that's an epimer at C2. The C2 carbon is different from glucose. And galactose is an epimer at C4. All right, know that definition of an epimer, difference of OH and H groups, and know that galactose is C4, mannose is C2, glucose is our blueprint. Not too bad? Different types of chair formations. There are different types of formations of our monosaccharides. Specifically, this one's talking about glucose. He said to not really worry about it, but just so you know, we can either have this line, this linear type way of drawing it, which is our Fisher projection. And if you think about it, like when you were in kindergarten, you were trying to draw a fish. This was the fish you made. So it kind of looks like a fish, Fisher projection. Our chair formation, if you turn this on the side and you're taking the right drugs, that can look like a chair. So our chair formation. And then we have our Hayworth projection, which is a circular, kind of looks like a halo. So Hayworth, circular, halo shape.
So you're going to show us pictures, or are we just going to discuss top of our head? Like for the epimers, so you're going to show us the pictures and say, where's the epimer at? You'll say C2 or C4?
I don't think so, but he could. I don't think so, though. I looked at y'all's test 1, and it was different than what I anticipated it being, to be honest with you. And I've heard from Try 2s as well that he's changed his Try 2 tests. So I think whatever it is about this trimester, he's changing stuff around about how he asks questions. Like he's still testing over the same material, but he's asking it in different ways.
Easier or harder way? Harder.
Yeah. And so he could put a picture on there. I don't think so, but he could. He just told me that y'all need to know this. Yeah. There's two cyclic forms of glucose. All it's saying is here at the end if the oxygen's on the bottom or on the top. I'm not going to ask you about that. Same thing with fructose. Ribose and deoxyribose sugars are used for nucleic acid synthesis. I asked it last week. I'm going to ask it again. What makes up a nucleic acid? Ribose sugar, phosphate, or pentose sugar, phosphate. Right. What are we missing here? Nitrogenous base. There it is. Nitrogenous base. Catch. Oh, ooh, nice. Nice. That's good. All right. Nitrogenous base, sugar, phosphate make up our nucleic acids. Sugars can bind to other sugars, and we've got a couple disaccharides here that we really need to know, and he's going to ask you about them. Boards is going to ask you about them. I'm studying this right now. Actually, I'm not studying this right now because I know it so well that I don't have to, which is what I'm trying to get y'all to do as well. So just trying to put this into long-term memory. We have lactose, which is right here. It's made up of glucose and galactose. So that one's pretty simple, right? Lactose is made up of galactose. All of these disaccharides have glucose in them, so you already know half of the equation already. It's just learning the other half. Lactose has galactose in it. Very, very, very simple. Sucrose is glucose plus fructose. All right? What's my thing here? Glucose is, they all have U's, so fructose has a U as well. So sucrose has glucose and fructose. So you want to leave that one behind. And then we have maltose, which is 2 glucose subunits on it. And that picture's over here at the bottom. Maltose. And I like to think of that as like I drink milkshakes, right? They're delicious, but they're really sugary. Glucose is our main sugar. Maltose is 2 glucoses.
We have to know the 2 beta, 1 beta, 4 stuff.
You shouldn't need to know that. Boards isn't going to ask that, so I don't think he's trying to teach this class so that you can pass boards. And his books actually do a pretty good job of preparing you for that. I'm going to be honest with y'all. Actually, a really good job. Polysaccharides. How many monosaccharides make up a polysaccharide? 7. More than 7, right? More than 7. I guess it probably could be 7 based, but if you ask y'all 7, put polysaccharides. 7 or more. They can be homo, where they're all made up of the same monosaccharides. That'd be like 10 glucoses in a row. Or they can be heteropolysaccharides, where it's like glucose and fructose, 7 or more. And we can make branches as well. All right, so we can have a straight line, or we can make branches. Just straight line, singular, linear, polysaccharides. Those are your alpha 1,4 linkages. All right, so that the first carbon on one molecule is going to attach to the 4th carbon on the next one. All right, and then 1,4, 1,4. It's just talking about which carbon it's attaching to. For alpha 1,6, whenever you see alpha 1,6, that's your branched chain. All right, so that's when we start making glycogen. Right? We start making glycogen so it's more soluble and that we can store more better. It's like being able to fold something up, like fold your sheets up like nicely and put it in there, right? Because we take that time to do that with our laundry instead of just letting laundry pile up. I'm letting laundry pile up right now. You can fold it nice and neatly as opposed to just letting everything just be one big clump and it'd be awful. Alpha 1,6 are your branches. Alpha 1,4 is just your straight lines. Glycogen, storage form of glucose in animals. Starches, storage form of glucose in plants. We have amylose and amylopectin. All right, need to know that. Here's a better representation of alpha 1,6, alpha 1,4, right? It's just between the first and fourth carbon. This is between the first and sixth carbon. Amylose is alpha 1,4. Amylopectin is both alpha 1,4 and alpha 1,6. Amylose starts with A. It's the shorter one, so it's alpha 1,4. Amylopectin starts with A. It's the longer name, so it has both alpha 1,4 and alpha 1,6. Okay? There's other polysaccharides as well. So greater than 7 or greater, different monosaccharides making them up. We have cellulose, which is beta 1,4, and chitin has beta 1,4 and acetyl D-glucosamine units. I doubt he'll ask that one. Cool. So cellulose, chitin both have beta. Amylopectin, amylose both have alpha.
And so basically, our bodies can't digest beta 1,4 linked polysaccharides.
That sounds right.
Cool. Let me know how the digestion cellulose goes.
Our amino sugars, I still haven't figured out exactly why. I haven't been able to get him to say what is on the test about these stupid things. But he said just the names? Cool. All right. Just know the names of these.
Just know that there's 6 possible amino sugars that can be used.
Cool. So be able to pick these out on a question. So know these names. Muramic, acetylmuramic. All right. So you really only need to know one right there. Glucosamine, glucosamine. So let's learn that one. Galactosamine and manosamine. So glucosamine, galactosamine, manosamine, along with muramic, acetylmuramic acid. Sounds fun. Look at that right before. Glycosaminoglycans, GAGs, we talked about it. Y'all talked about it, cells and tissues. We know it, just know hyaluronic acid, chondroitin sulfate, and know that it's repeating disaccharides making those. Okay, I don't think he's going to ask you about the names of those. Glycogen conjugates, it's just talking about what we can put a sugar on. We can put it on a protein, we can put it on a fat, or we can put it on a different part of a protein. Sugars attached by O glycosidic linkages and N glycosidic linkages. He told me that there's going to be a test question on this, and he said that y'all need to know which amino acids can participate in those types of bonds. Right? We have our O linkage is going to be amino acids that have a hydroxide on them. They have that oxygen on there. So that's what? Serine, threonine, tyrosine, right? And then our N-linked is going to be amino acids that have that nitrogen on there. So whether it's an amino group or an amido group, or I think it's the basic amino acids. Yeah. So it's hal, histidine, arginine, lysine, and then asparagine, glutamine that have that amido group on them. All right, so pretty simple. Just do you know which ones have an N? Pick that one. Which one has an O? Pick that one. And that's on page 24 for your reference. Questions on that? Not too bad? Still not too bad? Getting better? Let's say getting better. All right, why don't y'all take 5 minutes, palpate each other, practice that. Y'all keep honing those skills. All right, bringing it back. Carbohydrate metabolism. So we've learned about carbohydrates, right? We have mono, di, oligo, polysaccharides. We know how many of those have. We have learned how to classify them, right? Whether it's an aldehyde or a ketone, whether it's a triose, tetrose, pentose, or hexose. I still have to go through that every time I do it. We've learned how many isomers that they can make, right? How many different arrangements of those carbons, hydrogens, and oxygens we can make. We've learned that they're all D isomers, and we've learned how we can draw them. We've learned how they can be used elsewhere. We've learned what types of bonds they've had, whether it's alpha 1,4, linear, alpha 1,6, branched, beta 1,4, beta 1,6 in our starches, and amylose, alpha 1,4, amylopectin, alpha 1,4, alpha 1,6 also. We've learned that they are in our joints with hyaluronic acid, chondroitin sulfate, and that we can use oxygen and nitrogen bonds to link them as well. All right? That's chapter 8 in 30 seconds. Cool? Now we're going to learn how to break them down. Right? Hopefully we're not waiting until now to learn how to break them down, but if you are, good luck. All right. First thing we need to know is that we are oxidizing it. All right? We are taking glucose and we are oxidizing it. We are taking off those hydrogens and using those hydrogens to make ATP. So glycolysis and really any kind of metabolism of fuel is going to be an oxidation. Okay? End product of glycolysis, we're turning glucose into what? Pyruvate. Right? So at the end, so once we hit pyruvate, done with glycolysis. There's different, we can also go through gluconeogenesis, which is the new formation of glucose. All right, so that's taking, we're making glucose from non-carbohydrate sources. What molecule are we going to go through most likely to do that? Acetyl-CoA, right? Because it's the link between carbs, fats, and proteins. We can go through we have glycogenesis, which is the biosynthesis of glycogen from glucose. So that's if we have too much glucose, we don't need it, so let's store it as glycogen. And glycogenolysis, so we see that lysis, so it's the breakdown of glycogen releasing glucose. Know those definitions. You could easily ask one of those definitions on the exam. All right? But just read it. It tells you everything you know right in that word. It's just if you looked at it or not. Okay? Pentose phosphate pathway. I don't think he's going to ask it on this exam, but on a future one he will. But just in case he does, just associate it with NADPH and pentose phosphate. All right? It's a different way of breaking down glucose besides glycolysis. It goes through that pentose phosphate pathway, and it produces NADPH, pentose phosphates, PPP. Look for the P's. Okay? Glycolysis, oxidation. Utilized by all cells. All cells use glycolysis. Okay? We've talked about that ad nauseum. Glycolysis produces pyruvate aerobically. That was on my exam when I took it. It's an aerobic process. It happens in the cytosol. All right, glycolysis happens in the cytosol. So that ends. It's aerobic. It's aerobic. Actually, Google it. I talked to him at the half point of our class and after our class and the next day, and it is aerobic. If you see it on the test, is it anaerobic, aerobic? You better put aerobic.
Because it has the NAD involved.
If you actually Google it, I actually Googled it, and the first 3 things that popped up said it was aerobic, which was like really contrary to what I learned in undergrad. So don't on the test, he asked it, and I begrudgingly circled it, but I was circling 2 points. All right, so please just circle aerobic. You can grit your teeth while you do it and be like, I don't want to, but you'll get the 2 points. Okay? There's 10 enzymes in the pathway, what have you. At the end of it, we're going to make ATP, and we're going to reduce a coenzyme to make NADH. So we're turning NAD into NADH. And that NAD is a coenzyme, right, not a cofactor. Our cofactors are our what? Metal ions, right? And our coenzymes are our vitamins. Okay, glucose transport into the cytosol, right? So insulin is going to bring glucose into the cell, right? So it's going to take it right into the cytosol. First thing we're going to do is we're going to have hexokinase come in and put a phosphate on it, keeping it in the cytosol, right? So that phosphate that we put on it at the very beginning, first step we do is phosphate, keeps it within the cell, creates that polarity so it can't just go back out through. 2 phases of glycolysis. We have the prep phase, we have the payoff phase, and in the payoff phase, we are going to produce 4 ATP, net 2 ATP. So the end of glycolysis, we're going to net 2 ATP, we're going to net 2 NADH, we're going to net 2 water, and we're going to net 2 pyruvate. Okay? The water is less important, but we're going to net 2 water. Waters. Apologize. Proper English. All right. First thing we're starting off with is glucose. Right? We're starting off with glucose. Looking at the enzyme name, the enzyme tells us everything we need to know. All right. For those of you who've been here before, this will just be like further reinforcement. But for those of you who haven't, enzyme tells us what the substrate is with the prefix, and it tells you the type of the reaction with the suffix, and that's on page 49, I think, where it actually says that. Okay, so hexokinase, hexo tells us that we're dealing with a 6-carbon sugar, which is glucose, right? And kinase says that we're going to transfer a phosphate onto it, right? So we go over here, glucose gets turned into glucose 6-phosphate. So that 6-carbon sugar now has a phosphate on it, hexokinase. Okay? And the 6 just says that that carbon is on the 6th, or that phosphate is on the 6th carbon. So you can keep track of it that way. Next, we have phosphohexose isomerase. Phosphohexose, 6-carbon sugar with a phosphate on it. Isomerase, we're just going to make an isomer. Okay? So we go over here. Glucose 6-phosphate gets turned to fructose 6-phosphate. We go from a what to a what? Aldehyde to a ketone. Are you good? Did you have a question? Okay. All right, so phosphohexose isomerase. Pretty straightforward, right? Just dissect it down. It's going to make sense. Guarantee it. 3, phosphofructokinase 1. All right, we're going from, let's break that down, phosphofructokinase. Phosphofructose, there's a fructose with a phosphate on it. Kinase is what? Transfer of a phosphate, right? And we're going to put it on the first carbon, right? So we know our product's going to have a phosphate on the first carbon somewhere, right? And so we have fructose 1,6 bis phosphate, 2 phosphates on it. All right, it's that simple, that simple. What we need to know for hexokinase, phosphofructokinase 1, both of those are irreversible reactions. Okay? Both of those are irreversible because we're using ATP. Those are where we're investing our energy. Right? This is part of that investment phase. 1 and 3, irreversible reactions. Hexokinase, PFK1. Got to know it. All right. Next we're going to turn fructose 1,6 bisphosphate. We're going to split it in half and make glyceraldehyde 3-phosphate dihydroxyacetone phosphate. All right, so we're taking that 6-carbon sugar, breaking it into 2 3-carbon sugars, and that's with aldolase. So aldolase, you're thinking there's this big long name, fructose 1,6-bisphosphate. Screw that, that's way too long. Let's simplify it with aldolase, really simple enzyme, making 2 trioses. Then we have triose phosphate isomerase, triose meaning 3-carbon sugar with a phosphate on it, and we're going to make an isomer of it. So we're turning dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. So is that going from an aldehyde to a ketone or a ketone to an aldehyde? Ketone to an aldehyde. Good. Now we are in the payoff phase. We've got 2 G3Ps going through the rest of glycolysis simultaneously. That's why everything is in parentheses 2. So we're going to turn glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate with glyceraldehyde 3-phosphate dehydrogenase. Glyceraldehyde 3-phosphate, it tells us what we're starting with, not what we're ending with, what we're starting with. And we're going to dehydrogenate it. All right? So if there's a dehydrogenation enzyme, what are we most likely producing? NADH, like 5 times out of 6. Or, yeah, I think it's 5 times out of 6. Okay? What's the exception? Succinate dehydrogenase. You said the loudest. You get dum-dum. All right? So glyceraldehyde 3-phosphate dehydrogenase is where we make NADH. He could just ask you, what enzyme produces NADH? And you're just going to say the one that's in glycolysis that has dehydrogenase on it. It's the only one. Okay? He could throw in citric acid cycle enzymes in there to like throw you off if you don't know like which ones are where. So just pay attention to that. So you could have multiple dehydrogenases on there, but just pick the one that has G3P on it because that's pretty simple. Next, big step. We make our first ATP with phosphoglycerate kinase. All right? Phosphoglycerate, we have a phosphoglycerate kinase, transfer of a phosphate. Is transferring that onto ADP, making ATP, and we're left with 3-phosphoglycerate. Okay? That's our first ATP producing step. You could easily just ask that test question, which enzyme is the one that produces ATP first? Okay? 3-phosphoglycerate comes before 2-phosphoglycerate. And that's because we're starting off with 1,3-bisphosphoglycerate. And we just took off the phosphate on the first carbon, leaving us with the third carbon. So you got to know that 3 comes before 2. And we get to 2 with phosphoglycerate mutase. Right? Mutase, we're not making an isomer. We're not converting it from a ketone to an aldehyde or vice versa. All we're doing is we're taking that phosphate from the third carbon and putting it on the second one, just a mutation. Okay? Next, we have 2-phosphoglycerate to phosphoenol pyruvate, and that is with enolase. So this is kind of one of the few times where the enzyme does a better job of describing the product than it does the substrate. So phosphoenol pyruvate produced by enolase. If we are making water here, what type of reaction is that? Dehydration. All right, good. Dehydration type reaction. And all we're doing is removing that water to better set up the formation of this ATP. All right. So lastly, we have pyruvate kinase. Once again, does a better job of describing the end product. But there's also pyruvate here in this penultimate molecule, right? So we already know our end product of glycolysis is pyruvate. If we need to know what the second to last one is, we're probably going to pick the one that has pyruvate in the name too, right? Pretty simple. All right, pyruvate kinase, big step, irreversible, makes ATP, gives us our net 2 ATPs for the entire glycolysis, which makes it worth it. All right, if we didn't net, that second ATP, glycolysis would be not a thing because our body, evolution would have said, why are we doing this? It's a waste. All right? Who here does not feel comfortable with the enzymes of glycolysis? Because if not, I'm just going to skip ahead. Don't be shy. Who here feels 100% confident with the enzymes of glycolysis? All right. We feel good enough for a multiple choice test. Wonderful. It's a great answer. Good thing boards is all multiple choice. All right, well, I'll keep going through it then. What are the irreversible reactions? 1, 3, 10. Yes, the numbers are great. What are the enzymes? Hexokinase, PFK1, pyruvate kinase, right? Those are our irreversible ones. Right? Those are also our exergonic, highly exergonic reactions, right? They're releasing energy. They're also our rate-limiting enzymes, right? All three of them, yes, based on Dr. Sarkar's notes? Yes. Which one has a little bit more importance, though, than the other two? PFK1. And what is special about PFK1? We haven't quite covered it yet, though. It's the first committed step. All right, so PFK1 is the first committed step. Okay? I'll throw a sucker that way. Don't be, off the wall. Meant to do that. Awesome. All right. PFK1, first committed step. We'll break down each of the steps of glycolysis here in a second. But pyruvate can go one of two ways. Okay? Pyruvate can, pyruvate can go on to form acetyl-CoA with what enzyme? PDC, pyruvate dehydrogenase complex, in humans, right? So we can either, if we continue this aerobic condition, pyruvate can go make acetyl-CoA. In anaerobic conditions, in animals, it produces lactate, okay? So it's fermented to lactate, and this is going to be like in rigorously contracting muscles, right? So like sprinters or power lifters, right? They're going to use up, they're doing things that they're going so fast, they don't have time to go through aerobic processes, so they just use the anaerobic, which is faster, but you're going to make lactic acid as a result, causing your muscles to hurt a lot, like when I played basketball last night. Yeah, don't play clinic, okay? They're good. Yeah, I think they made their first 13 shots against us, so it was like 35-7. It was a good day. Yeah, they have a little bit more free time, but they're just really good. All right, so we can make lactate in animals with fermentation in anaerobic conditions, all right? In yeast, though, pyruvate can be converted to ethanol through fermentation. All right, so in yeast, we get ethanol and carbon dioxide, whatever, fermented to ethanol in yeast. Need to know. He's going to ask you something about that. All right, first step, hexokinase. Right, what does hexokinase convert glucose into? Glucose 6-phosphate, okay? Glucose 6-phosphate. That enzyme requires magnesium as a cofactor, coenzyme. Cofactor, all right? When in doubt, if he asks you what cofactor makes an enzyme work, magnesium, all right? Magnesium. So glucose to glucose 6-phosphate is hexokinase. Hexokinase is present in all tissues, right? It's present in all tissues. Hexose just means 6 carbon. You will find out later that hexokinase actually works on fructose, which makes sense, right? Because it's also a 6-carbon sugar. So that increased versatility of hexokinase versus just straight glucokinase is why it's present everywhere. And glucose kinase is more specific, so it's present in the liver only. Okay? So glucose 6-phosphate. If you are unsure about these, write these down as we're going. Write it out. Get used to doing that yourself as opposed to just hearing it. Add that extra kinesthetic aspect to the learning. Okay? 3 major purposes of making glucose 6-phosphate. The biggest one to me is that once you put that phosphate on, that glucose isn't going to leave the cell. It traps it within that cytosol, right? Glycolysis takes place in the cytosol. That's a board's question. You think it's pretty simple. It's a board's question, right? People think biochem on boards is going to be like the worst thing ever. It's not that bad, right? Pay attention now. Do the work now so you don't have to do it later. The biggest one is the number 1. Look over 2 and 3. The biggest one is number 1. Step number 2, enzyme is phosphohexose isomerase, right? Phosphohexose isomerase. Breaking it down, phosphohexose, there's a 6-carbon sugar with a phosphate on it. Isomerase, we're going to make an isomer of it. So we're going to turn glucose into fructose. Is fructose an aldehyde or ketone? Ketone, right? It seems like I'm asking the same question over and over again, which I am, but right, it's not that hard. It's not that hard. So glucose 6-phosphate into fructose 6-phosphate. Cofactor, look again, magnesium, right? It's going to show up. This step is, nope, that's the next enzyme. The next step is PFK1, our first committed step in glycolysis, phosphofructokinase 1, first committed step. We use another ATP. So in hexokinase and PFK1, we're breaking down ATP to make that happen. It's the investment phase. Okay, we're turning fructose 6-phosphate into fructose 1,6 bisphosphate. Right, bis just means 2. Okay. Fructose 1,6 bisphosphate. We've got that second phosphate on. One's on the first carbon, one's on the 6th carbon. So we're going to break it in half so that we have 2 3-carbons with 3-carbon sugars with phosphates on them with aldolase. All right. Aldolase is going to make glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Both of them have phosphates on it, right? So we can see that every step in glycolysis has a phosphate on it. I guess except for pyruvate. I guess we got rid of the phosphate at pyruvate, but whatever.
Is pyruvate considered an intermediate, or is it just all the steps between like the first and the last steps of the intermediates?
I would say the intermediates of glycolysis would be the steps between.
Okay, so it'd be everything besides glucose and pyruvate would be an intermediate?
I would think so. That's what you start and end with. I would think so. But if Dr. Sarkar has said something else, then I would defer to what he has said. All right, so we got aldolase making dihydroxyacetone phosphate, glyceraldehyde 3-phosphate. Not that big of a deal. They're isomers of each other. So we're going to, instead of trying to take glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, I'm going to keep saying it just to get that in your head, what the words are. Instead of taking both of those things and doing their separate processes, let's just say, screw it, let's just turn dihydroxyacetone phosphate into G3P. Call it good. All right? So that's the next step, triose phosphate isomerase. Triose, meaning what? 3-carbon sugar with a phosphate on it, right? And isomerase, making an isomer, pretty simple. Ketone to aldehyde. Ketone to aldehyde. All right, step 6. Now we're in the payoff phase. So enzyme number 6 is where we first start making something. Say again? Triose phosphate is 5. That's 5. Yes, glyceraldehyde 3-phosphate dehydrogenase is going to turn that glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate, producing, big thing here, NADH. What step produces NADH? The dehydrogenase one, G3P dehydrogenase. All right? Let me just check something real quick here. All right, that's a reversible reaction. That's not that big, though. All right. And that's an oxidation type reaction. So it's oxidation of aldehyde to acid. He could ask you the types of reactions in glycolysis as well. So this one is an oxidation. Whenever we see the NADH, it's either a dehydrogenation type reaction, or in the case of citric acid cycle, we're also releasing the CO2, so it's a decarboxylation. But we can get to that. And whenever we're dealing with enzymes 1 or 3, those are phosphorylations, right? We're using phosphates. Substrate level phosphorylation, step number 7. Right, this is the first time we make ATP. Big deal, right? We got our money back. So that's going to take 1,3-bisphosphoglycerate and make 3-phosphoglycerate. We've lost a phosphate, right, because we made ATP. Look at that. Magnesium is the cofactor. What do you know? All right, one of the most highly supplemented metals in our diet because we need it for all these different types of enzyme processes. All right, now we've got 3-phosphoglycerate and some ATP. We're going to turn 3-phosphoglycerate into 2-phosphoglycerate. All right, so 3 to 2, that's with the mutase, right? So what is the substrate for phosphoglycerate mutase? 3-phosphoglycerate. All right, so he could ask you that way. What is the substrate of this enzyme, or what is the product of this enzyme? All right. So now we're at 2, and look there, magnesium, perfect. All right, this one is that is not where we produce water. That's just a reversible shift mutase. He's probably not going to ask what type of reaction that is. Dehydration to form pyruvate. All right, so step 9 is we are turning 2-phosphoglycerate into phosphoenol pyruvate. That requires magnesium, but it can also use manganese. All right, can also use manganese. This is the first time we've seen something different. So he could ask you that. Probably not, but he could. It's a dehydration type reaction. We're losing water. Lastly, substrate level phosphorylation, making ATP, going from phosphoenol pyruvate to just pyruvate, right? The pyruvates line up, makes sense. Pyruvate kinase, we make our second, or I guess our third and fourth ATPs from glycolysis, net 2. Great.
Will he refer to the steps by their numbers, or will he actually explain like the names of the stuff?
He's going to have the names. He won't have the numbers. Okay. The numbers are just kind of for you to kind of have an idea of where you're at in the cycles. All right. He's not going to ask you about the energies, so don't worry about memorizing the numbers as far as I'm aware. But just know that the exergonic couples to endergonic to make those breaking of bonds correlate with the making of bonds. Glycolysis can provide ATP in the absence of oxygen, right? So this is what we're talking about with aerobic, anaerobic, and that's through the substrate level phosphorylation. It can happen, but for all sakes purposes, glycolysis is aerobic. Clinical correlates, mature red blood cells lack mitochondria. That's a big thing later, but also now. So red blood cells, they don't have the mitochondria. That's where we have our oxidative phosphorylation after the citric acid cycle, right? So if they don't have that ability to generate ATP via hydrogen carriers like NADH, FADH2, then they can only go through glycolysis. So that's all they can use to get energy. So our red blood cells, in order for them to keep living and help bring oxygen to the rest of our bodies, has to use glycolysis. Aerobically, we're going to take pyruvate and make acetyl-CoA, and that's with what enzyme? PDC, pyruvate dehydrogenase complex. Need to know that enzyme name, need to know what it does, pyruvate to acetyl-CoA, and you have to know the 5 different factors that go into making that enzyme work.
Do we need to know the 3 enzyme that makes it up?
No.
No?
As far as I'm aware, you don't need to know the enzymes that make it up. As far as I'm aware. Boards, I don't think boards cared. Boards just wanted us to know the name and the things that make it work. All right, so those are, we're going to start with which ones? We're going to start with the hard ones, right? So TPP, lipoate. All right, say those first. Work on saying those first so you don't forget them. They're the hardest ones. All right, TPP is, thiamine pyrophosphate. All right, that's one of your B vitamins. That is always present when we have decarboxylation reactions. All right, so whenever we're losing CO2, TPP is right there to make that enzyme work. And then we have lipoate, and then the easy ones of NADH, or NAD, FAD, acetyl-CoA, or CoAsh. TPP, lipoate, NAD, FAD, CoAsh. Gotta know those five. Gotta know those five, gotta know those five, gotta know those five.
Well, we have to know that like CoAsh is vitamin B5, and like NAD is niacin.
He said it a couple times.
You said no, TPP is D1, and I think that's pretty much it.
Okay. I know in Biochem 2, like he's going to hammer those enzymes, like y'all learn all those enzymes and deficiencies and all that stuff in Biochem 2. From what I remember when I took the class, he didn't list it by the number. He listed it by the name of like TPP or lipoate, but if he has hammered it, if he has said it 2 or 3 more times, then just know that TPP is 1, NAD is what, 9? Niacin is 9 or 6?
TPP is B1, CoAsh is B5, NAD plus is niacin, FAD is riboflavin, and lipoate is made in the body.
Cool.
Which one's B6?
B6?
Don't worry about B6 if it's not one of the B numbers. It gives off FADH2. Yes. So that FADH2 would be should be a product. Should be. Just trying to see if I can find it down here. NAD, NADH.
I thought I heard him say that I should make sure that
It should be. Perfect. Oh, actually, no. FADH2 is not produced in this last one. It says the electrons pass first from FAD, and then that gives it off to NAD. So it's like a baton carrier. So FAD just helps with the formation of NADH2. NADH. So FADH2 not produced. Sorry for that confusion. All right. Anaerobically forms lactate in humans. Pyruvate can be fermented to make ethanol in yeast and bacteria. Our regulatory enzymes, hexokinase, PFK1, pyruvate kinase, need to know those 3. They're the irreversible ones, highly exergonic, deal with ATP. All right. PFK1 is the first committed step. Need to know what activates it or turns it off. So hexokinase is inhibited by glucose 6-phosphate, right? That's just the negative feedback. So if we have a bunch of this product, stop making more product, turn off hexokinase. PFK1, inhibited by ATP, right? We don't need to make ATP if we have it. And citrate, citrate's the awkward one. We haven't had that yet, but it's in, it's pretty much, it's feedback from the citric acid cycle. If we have citrate, then we don't need to be making more. And it's activated by not having ATP, and fructose 2,6-bisphosphate, which you will learn in Biochem 2, and ribulose 5-phosphate also activates it. Also Biochem 2. Pyruvate kinase, irreversible, exergonic, inhibited by ATP, right? Energy inhibits. Acetyl-CoA inhibits, right? That's like the end product after pyruvate dehydrogenase complex. Long-chain fatty acids will also inhibit it. And that's chapter 9. All right, one more, really quickly.
Andrew,
Yes, sir.
can you explain why ATP would inhibit phospho or PFK1?
Because we're going through glycolysis because we need energy, right? So if we already have it, why are we going through it? That's why. Citric acid cycle. All right, we're taking that acetyl-CoA that we made with pyruvate dehydrogenase complex, TPP, lipoate, NAD, FAD, CoAsh, and we're going to put it into the citric acid cycle. I do not believe that pyruvate dehydrogenase complex is a part of the citric acid cycle. I think the citric acid cycle begins with acetyl-CoA combining with oxaloacetate, just to kind of clarify that. It's in pyruvate dehydrogenase complex is in the mitochondria. Need to know that. It's in the mitochondria. Need to know the 5 coenzymes. Less important is the 3 other enzymes that make it up.
Do we have to know what each of the 3 enzymes are?
As far as I'm aware, no. As far as I'm aware, no. We didn't when I did it. Last try, I don't think they needed to either. She asked if we needed to know how these work, but I doubt it. Focus your attention elsewhere. No.
Andrew, you said before, you said the electrons pass first
So
FAD. So wouldn't that mean it does actually make FADH2?
net, no. So it makes FADH2 in the middle of it, but then it's like the middle leg of like a relay. Then it gives those hydrogens off to NAD, making NADH. So an end product is not FADH2. The end product is NADH. So test question, is FADH2 produced by pyruvate dehydrogenase complex? No. Look over this real quick. We're running out of time. Look over what inactivates it, what activates it. It's pretty much the same things. End products inactivates it, and anything that is not energy is going to activate it, or if it's a coenzyme. Phosphorylation inactivates pyruvate dehydrogenase complex. When you take off that phosphate, we activate it. All right, so we can turn it off by putting a phosphate on, and then it's kind of like a boot, like a car boot. You can't drive your car if that boot's on, or like the steering wheel stopper, clamp, whatever thing that's called. Then we take it off, it can work. Citric acid cycle occurs in the mitochondria versus in the cytosol for glycolysis. We make 3 NADH and 1 FADH2 in the citric acid cycle. Okay, also 1 GTP. So those are the 5 big things that we're making with the citric acid cycle. There's a couple other smaller ones, less important. Very, very important that you know for the citric acid cycle the types of reactions. All right, he has them listed here. You need to know what those are 100%. So outside is the type of reaction. On the inside, we have our enzymes, and then in the middle of that, we have our molecules that we're making. First thing we do, acetyl-CoA. It's a 2-carbon sugar combines with oxaloacetate, making citrate. All right, so citrate is where we start, and that's with citrate synthase. That's a condensation reaction. All right, so citrate. When I learned this, I kind of just had the general idea. I saw this as a clock. Right? I just tried to have a general idea of where things were in relation to that clock, of that circle, just to kind of have something to fall back on. But if you are like a mnemonics person and you need like the mnemonics, just look one of those up, quick Google search. That'll give you everything you need. Make it dirty, make it clean, whatever helps you the most. So we're at citrate. Condensation reaction. Citrate. Citrate then goes through a dehydration and hydration reaction to make isocitrate. So that makes pretty good sense that we're going to go from citrate to isocitrate. That enzyme is aconitase. I don't know how that is named that way, but it is aconitase is enzyme number 2. All right, those are dehydration, rehydration reactions, or hydration type reactions. So we're at isocitrate. Next enzyme is isocitrate dehydrogenase. What do you think we're making with that, based on that name? NADH, right? All day, every day. We're making NADH. Okay, so boom, that's where our first NADH is made, isocitrate dehydrogenase. What's special about this enzyme, though, is that it also breaks a carbon-carbon bond, producing CO2. All right, that CO2 takes more precedence in the naming of that type of enzyme than does the production of NADH. So that is why we are naming this oxidative decarboxylation. We are removing the carboxyl group. Okay, and that makes alpha ketoglutarate. Alpha ketoglutarate. All right. Alpha ketoglutarate is then converted into succinyl-CoA via alpha ketoglutarate dehydrogenase complex. All right. It's the exact same reaction, same type of reaction as isocitrate dehydrogenase. All right. It produces an NADH. It gives off a CO2. If it gives off a CO2, that's going to be what kind of enzyme? Oxidative decarboxylation. All right, and that's going to have TPP as its coenzyme, right? What we saw from thiamine pyrophosphate from pyruvate dehydrogenase complex. It's the same TPP that helps produce that CO2, which is yes, yeah. He's saying like step 3. He's saying that both isocitrate dehydrogenase and alpha ketoglutarate dehydrogenase use TPP because they're decarboxylations. Now, we are at succinyl-CoA. Succinyl-CoA. CoA just reminds me that I'm looking at a clock. We're at 6 o'clock. All right? Whatever. I try. So we have succinyl-CoA. We're going to turn that into succinate. All right? That enzyme is succinyl-CoA synthetase. It's talking about succinyl-CoA, talking about the substrate. Synthetase is the breakdown of it, pretty much. And we're making succinate, but the big, huge thing here is we are making GTP. All right? Succinyl-CoA synthetase is where we make GTP. All right, you can see where I'm emphasizing the T there. Question? That would make sense. I would believe so, yes. We have our three regulatory enzymes for the citric acid cycle are citrate synthase, isocitrate dehydrogenase, and alpha ketoglutarate dehydrogenase. Those are our big regulatory enzymes, are those 3. I don't think he's going to ask if those are exergonic or not, as far as I'm aware. So synthetase produces GTP. Synthetase GTP. Next, we're going to go from succinate to fumarate, and that's with succinate dehydrogenase. It's talking about the substrate, dehydrogenase. All right, this is the one time that dehydrogenase does not mean NADH. All right, this time we are making FADH2. All right, it would suck if you forgot that this was the only one that made FADH2. All right, succinate dehydrogenase. You already know the dehydrogenase. There's going to be some kind of removal of a hydrogen. All right, so don't let it suck and get this one wrong by not putting that this one produces FADH2. All right, big. It's the only one that's different, so you know he's going to ask about it, right? Okay. That's a dehydrogenation type reaction. And we'll go back and compare that versus over here after malate. Next, we're going to take fumarate and convert it into malate with fumarase, right? We're breaking down fumarate with the enzyme making malate. That is a hydration reaction. And next we have malate dehydrogenase, taking malate, converting it back into oxaloacetate. That is a dehydrogenation reaction because we have a dehydrogenase, right? So now we see a dehydrogenase, we're thinking production of NADH, right, which is exactly what it does. That's our third step that makes NADH. Real quick, on the bottom right-hand corner of our clock, right, we have isocitrate dehydrogenase, alpha-ketoglutarate dehydrogenase, right? They produce the NADH, but more importantly, not more importantly, in the naming of it, more importantly, they produce that CO2. So these are oxidative decarboxylation. Over here with succinate dehydrogenase and malate dehydrogenase, those produce NADH and FADH2. That's all they do. So they are categorized as dehydrogenation only type reactions. Okay? It's really, really important to be able to differentiate between those four. So the ones on the left are dehydrogenase. The ones on the right are oxidative decarboxylation. Needs to know that because the test question would be, which of the following is a dehydrogenation reaction? And he'll put all four of those dehydrogenases on there. And he'll say pick two. You don't have to know which two to pick, right? And it's just the succinate and the malate. Not that bad, right, if you've studied. And that is pretty much the citric acid cycle. Irreversible ones, like I said, citrate synthase, irreversible, highly exergonic, rate regulatory, alpha-ketoglutarate dehydrogenase and isocitrate dehydrogenase, also irreversible, highly exergonic, rate regulatory, both of them. All right, so those three are rate limiting, highly exergonic. Don't forget succinate dehydrogenase, please, 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 please. If we get 100% on that question, that'd be awesome. There's never 100% on that question. All right, just like when y'all did the ileum, right, for Dr. G, right? People missed it still, right? Yes. Don't be the person that misses this one. That's the same level of importance, in my opinion, as ileum for DAA. What produces FADH2? Succinate dehydrogenase. Yeah. All right, big here. I've seen this on each of the last tests. We have NADH. 1 mole of NADH produces 2.5 ATP. 1 mole of FADH2 produces 1.5 ATP. All right, we're producing more NADH. All right, we're doing that because it's a higher currency. All right, we can get more out of it, so we're trying to produce more of it in the citric acid cycle. So NADH produces 2.5, FADH2 is 1.5. All right, that's just trying to get the most bang for our buck with the citric acid cycle. Rate limiting, talked about it. Talked about that. Whatever. Just read through this really quickly, specifically the feedback inhibition, allosteric regulation. Just going to try to finish on time. Anabolic reactions. So anabolic, are we making or breaking bonds? Making, right? So we can take some things from the citric acid cycle and make other things with it, with gluconeogenesis, amino acids, porphyrin synthesis, which is going to be part of your hemoglobin. Wow, okay, we just had that test today, and it makes sense, and it's awesome. Lipid biosynthesis, where we have our catabolic, we're breaking stuff down, and we can make carbs, fats, amino acids. Biggest thing is anaplerotic reactions. Need to know that definition. Refill TCA cycle intermediates that have been extracted for biosynthesis of biomolecules. So whether it be malate or fumarate or what have you, has gone on to do those other processes that we just talked about we need to get those back into the citric acid cycle so that it can keep going. So that's what anaplerotic reactions do. They just try to fill in those gaps where they went off and helped somewhere else. Yeah.
Is it primarily pyruvate that is the one that helps out? It looks like in this reaction, pyruvate's the one that puts oxaloacetate back in. Pyruvate's the one that puts malate back in. Phosphoenol pyruvate is the one that puts oxaloacetate back in. Is it primarily pyruvate that does that?
I have no idea, to be honest with you. I have no idea. Could be. Also, down here, take a look at these enzymes that are in blue and take a look at what they do. Pyruvate carboxylase takes pyruvate, makes oxaloacetate. He's going to ask you what enzyme does that, if anything. So just 4 more enzymes. I know we just learned, like 17 of them. Just 4 more really quickly. Those should be a lot more straightforward. And that's 5.5 chapters in 2 hours. Questions? I think the tissues are less important than knowing the enzymes and their reactions. I would learn those first. Any other questions? Yeah.
So I looked up some mnemonics, and some of them started the products with O, and the other one started it with C. So does it for sure start with O? Because you said O, oxa, whatever.
The first step of the citric acid cycle is taking oxaloacetate and acetyl-CoA, making citrate.
Okay. So it does start with O. Okay.
So I would say the first enzyme is citrate synthase, making citrate.
Wait.
So citrate, I said the same thing, just 2 different ways for them. So you're starting off with oxaloacetate and acetyl-CoA, right? Those are going to combine to make citrate via citrate synthase. So I would say start with citrate. But you could just ask him right before the test tomorrow to see, hey, like what is like the first thing? Because this is just every time we're going through glycolysis, we are regenerating oxaloacetate to go back through it again to combine with acetyl-CoA.
2 more questions. Okay, so when molecule inhibits the action of